Okay, hello, and thanks for the opportunity to speak. My name is Gilbert. I'm presenting an abstract presented by my students at the ISMRM this year. It's about our tries to extract metabolic profiles from 7 Tesla MRsi to classify tumors according to the grade and IDH mutation status. First of all, I have no financial interests or relationships to declare, and I want to introduce our work a bit better. So when I'm saying we, I'm mostly speaking about the Medical University of Vienna in general, and specifically about the 7 Tesla scanner we have at the Highfield MR Center, and now the 3 Tesla interoperative MRI we're running since last year, which of course requires a lot of cooperations, neuroradiology, oncology, histopathology. So many people are involved in this study, so it's very hard to address all of the works adequately. But this is now mostly about my students Cornelius and Sukrit, who ran a lot of statistical evaluation and ratio extraction and other stuff of our MRsi data. When I'm speaking of MRsi, I normally refer to the combination of magnetic resonance spectroscopy that in single voxel extracts the spectral information instead of an image in one volume, while in imaging, of course, we acquire spatial distribution about a whole slice or 3D volume. In MRsi, we need to combine this, which historically has led to the issue that we need to acquire a lot of signal in every spatial volume, and that requires some time to get the spectral evolution. And we want to do that with as high resolution as possible, and we want to do that as fast as possible. So at our center, we have developed methods in cooperation with the other departments for at least a decade. And we want to use that in neuro-oncology specifically because we think, or literature says, that there are many interesting points of the tumor metabolism that proton MRsi in the human brain can access. I highlighted some in red here in this image. 2-HG, so 2-hydroxyglutarate, has been the buzzword over the last decade because it is directly related to IDH mutations. But there are also other markers like glutamate and glutamine or glutathione or lactate that are, depending on the method, a bit easier to image. And we have to go down the profiling route of the other stuff anyways, because at least our method is not really sensitive to 2-HG. I have to admit, if we try to fit 2-HG, we get more 2-HG fit outside of the tumor or in healthy brains than in the tumor, so I'm pretty sure it's not working. But what works is the imaging of some other metabolites. So as you can see here in this image, we have images. We have a high-grade glioma where we can see, in comparison to the contrast enhancement specifically, that we can see different anatomical different tumor structures resolve differently in anatomy. If you look at the glutamine and the colon maps, with colon being the classical marker that is used in neuro-oncology, that we see differences in the distribution of our heterogeneous hotspots between colon and glutamine. And we think that someday we can really extract local profiles. Also, a highlight here would be glycine, which is only with great effort separable from inositol in the next row at lower fields. You need specific methods for that might or might not work. We think we can separate it very nicely at the high fields. And if we would have this glycine and the myoinositol together, you would not see anything special, or you would not see the glycine at all. And if we extract the glycine, we have this very nice hotspot there too. And also you see with glycine in the healthy brain, you still at 7 Tesla cannot really fit it at all. The concentration is under 1 millimolar. So that's one of the things that you only see really in the tumor. So for this work, we started one easier step. We said our goal was to determine IDH status and grade for a patient. So not do specific nosologic maps or TME maps, but really start with something that would be already of clinical interest, IDH status detected preoperatively. For this, we included all the viable patients we had at that time, so 24 high-grade gliomas and 12 low-grade gliomas, quantified did the MRsi with LC model, and to have a control volume, Julia did the segmentation of all the tumors, which was a lot of work. And you can see in this upper image that the segmentations could then be already visually compared to the MRsi in this case. So we see that we don't have a colon hotspot in the necrosis, which is good to know. And now my students progress to statistical analysis in R and later in Python. So to extract all these metabolite maps, calculate ratio maps, and then try to do voxel-wise analysis. So first for this work, it's mostly about calculating the ratios of all these metabolites and then look at the distributions and see if anything comes to our mind that would effort further research. And first of all, we have the nice control volume. If we look at tumors versus normal appearing white matter, we found that we would see increased medial voxel values for a lot of these metabolites. So as we can see from the maps, there's a lot of stuff that changes. Also some go down like NAA or inositol. And at the ISMRM time, this was basically the state we have since refined the model, as we'll show a newer result later, that if you then go to the distribution over all these patients, things start to look less fancy. So while we have these very nice hotspots, if you do these ratios and then look at the statistical distributions, you still see that the range is incredibly wide. We observed that especially for glutamine, some patients have really, really high volumes in the ratio to NAA, like in the range of glutamine to NAA of 10. Some are way lower, and our cohort is not big enough to really see if there's some specific mutations behind that. Also, for IDH, we found a few things that also might be investigable, but to point out the big problem that we currently have and that we want to address with future research is that ratios are, of course, you see here, if you want to develop models that are all some ratio to creatine or NAA, they are, of course, not independent of each other because you have some common divisor in all of these. But right now, we don't have the technology to do quantitative analysis in each tumor voxel. So we have to use a lot of parameters that are not independent of each other, or you easily produce artifacts like, let's say your NAA decreases in the tumor, your hotspot increases by the division, but at some point you don't get an NAA fit at all. You divide by 0, you have to exclude the voxel. So maybe the most interesting voxels you cannot even include with that. As you see, this is causing that some of these results here are not that interesting for this stuff. Now we are working on improving it, and one of the steps was separating the tumor really in the regions. We want to look at the comparison normal appearing white matter, edema, non-contrast enhancing, contrast enhancing, necrosis, and also the peritumoral region that could already, so peritumoral means the stuff around the clinical segmentation that is not morphologically active, but could have some kind of altered metabolism. So this is a very preliminary analysis where we still see in the contrast enhanced regions over all tumors, we would really start, so of course contrast enhancement means it should be the high-grade ones, that there is a difference between IDH wild type and mutation that looks like you could derive some classifier. And the classifier work is the one my students right now are doing, and we have first results that lead to, with a combination of ratios, some accuracy of over 75%. You know, models can be trimmed anyways. We try to do something that is statistically feasible, but also our sample size is very limited. But we hope that at some point we can really extract useful information. And our thought is we have more metabolites than are available with the clinical methods that can mostly do creatine, choline, and NAA. So we have more metabolite information. And with the higher resolution, we have more voxels. So you could try something like classify every voxel in a patient and then look at the majority report of this patient and say 70% of the classification, say IDH, then you classify it as IDH or something, which would make us a bit more robust to artifacts. Because we only have one voxel or 3 voxels in a patient, it really depends of the fit of that voxel. If we have 1,000, it doesn't matter if 30 of them are really bad or don't work out, because the statistics should work for us. But we are currently in the process of doing that. With that, I'm coming to an end. There are some supplementary data actually in the internet available for these tumor patients if you want to look at them really at the maps themselves, or you can contact me per email or talk to me now here at this meeting. I'm open for all questions and so on, and I want to thank all my colleagues and all the funding agencies funding our work. Thank you.